Okay, so we finally have our egg and sperm. That's what the whole entire last chapter was about. How do we go from an egg and a sperm to an entire new human being approximately 9 months later? Well, the egg and sperm have to join up and combine their DNA, so that process of fertilization has to occur. From there, we have a lot of stuff that has to happen. So that fertilized zygote has to make its way, implant itself into the uterus, and then it has to grow and develop. We have to have a placenta to provide it with all of those nutrients. And from there, we have to go through a process called parturition, which is actually the act of giving birth. And there are a whole lot of structures that have to develop as that happens. So let's start to look at the very first step, which is fertilization. A few terms before we jump into things. The conceptus is what you can call the developing tiny human at any point in this process. So at whatever stage, you can always call it the conceptus. Gestation period is the time from ovulation to parturition. So we call that, the whole thing pregnancy, and that's about 280 days long. The embryo is what we call the developing new person from 3 to 8 weeks of development. And then after that, it's called a fetus from 9 weeks up until the time of birth. Before that, it's going to be called a blastocyst and a couple other things, and we'll look at what those individual structures are. But let's talk about fertilization first. So to get a new tiny human, you obviously have to have a sperm and an egg. We already know how we get those things. And this fertilization process has to occur probably somewhere within the uterine tube of mom's reproductive tract. So the oocyte, once it is ovulated, wrapped up still by some of those granulosa cells, so it can survive in the uterine tube for about 12 to 24 hours. But after a day, your chances of getting pregnant are basically nothing. Sperm can last quite a bit longer, about 12 to 48 hours. So you have a window of fertility. So you can, have sex a day before you ovulate and probably still get pregnant. But after you ovulate, the next day, your chances are basically nothing because that oocyte will no longer be viable. If fertilization does occur, then we form this thing called the zygote. That occurs after you have physically combined the DNA of both the egg and the sperm. So once their nuclei combine and you have one nucleus with all of that genetic information, you can call that finally a zygote. That's still just a single-celled organism, still has to develop into an embryo and then into a fetus and so on. All of the sperm that are not successful in that process, there are millions of them, have fate somewhere along that track. So they can leak out immediately after sex. They can be destroyed by the acidic environment of the vagina. They can not pass through the cervix, even though the semen contains some enzymes to help loosen that barrier. And then in the uterus, there are going to be a lot of phagocytes present that if they come across the sperm, they will destroy them. And so only a few thousand actually make it into the uterine tube. You maybe have a couple thousand that actually get to the egg. They have to swim up the uterine tubes, still makes it the final few inches. But you need a lot of sperm to actually make it to the egg. So you can't just have a new baby from one single sperm because a lot of them, millions are going to die before they even get to the uterine tube. And we need a bunch of them to actually make it all the way to the egg, which is what you see in this picture here. So this is the oocyte surrounded by a bunch of sperm. And, all of these sperm are necessary to go through this process of trying to dig through the layers of that egg to actually get their DNA in. You need a bunch of them because the egg's super gigantic and the sperm are teeny tiny, and they need to be able to kind of like help each other out getting through that surface. So once sperm are in the female reproductive tract, there is, quite a bit of stuff in the female reproductive tract that are going to start to degrade the acrosome at the tip of that sperm head. So you have the acrosome, which is the tip on top of the nucleus, and that acrosome contains the enzymes that are actually going to burrow their way into the egg. But you need to loosen the membrane around that acrosome before that sperm can actually release its enzymes. So that's called capacitation. So that process has to occur, this thinning of the membrane surrounding the acrosome so that their enzymes can be released onto the surface of the egg. The sperm have to continue to swim up the uterine tube to meet the egg because the egg is traveling super slowly. So they can't wait. They need to travel to the egg. Once they're there, it takes a bunch of sperm, like thousands of sperm, to actually penetrate the egg. And that requires something called acrosomal reaction. So that acrosomal reaction is when they actually release their enzymes out of the acrosome near or on the oocyte, and that is going to start to penetrate the outer layers that are surrounding the oocyte. So hundreds, if not thousands of them are going to be necessary to start breaking down that barrier, and that's what's happening in this picture right here. You have a whole bunch of sperm doing the exact same thing, releasing those enzymes, trying to burrow their way somewhere into this egg to release their DNA inside. Ones that arrive on the scene later are more likely to actually fertilize the egg because they don't have to do any of the hard work, and the sperm are going to, not survive this process if they're working at it for too long. So the ones, arriving to the scene after these holes have already been started are more likely to fertilize the egg. This is a cool closer up picture of this process happening. So the sperm are actually going to bind to the oocyte surrounding it, and they are all secreting these acrosomal enzymes trying to break it down. So as the sperm approach, I didn't talk too much about these layers in the last chapter, but we have these granulosa cells out here that are still present. Even though this oocyte has been ovulated, some of those granulosa cells are still present. The rest of them were left behind and became the corpus luteum. But interior from this granulosa cells, these are called the corona radiata. So that's what those granulosa cells are. Interior to that, you have this blue layer. That's the zona pellucida. That's the layer that the sperm have to dig through to try to get to the oocyte. So they have to dig through that outer layer, and it takes a lot of them to do that. Some of their enzymes are going to be used just burrowing their way through these granulosa cells, but those are, more loosely connected, so they can kind of more easily get through there. They will need to burrow through this entire zona pellucida and then make their way to the plasma membrane. So this inner layer here, this tannish layer with those receptors that you can actually see on them, that is the outer plasma membrane of the oocyte. The zona pellucida has these receptors, all these little yellow dots. There are also these receptors on the plasma membrane. So these sperm have to bind in two separate places before they can actually fertilize the egg. So this is what it looks like when we have this acrosomal reaction beginning to happen. So these sperm are going to bind on the zona pellucida after they've dug their way through those granulosa cells. Once they bind to the zona pellucida, there are these binding sites, those little yellow dots. There is going to be an influx of calcium into the sperm. That is going to cause the release of those acrosomal enzymes. Remember, this only happens if capacitation has occurred and that acrosomal cap is thin enough for that to happen. So after they bind, you have the acrosomal enzymes being released. One single sperm is going to make it through here and bind to one of the receptors on the plasma membrane of the oocyte. So it's bound to these receptors just to start to release its acrosomal enzymes. Once it binds to one of the receptors on the plasma membrane, then we have this extremely important reaction occurring where you have the fusion of the plasma membrane of the sperm and the plasma membrane of the oocyte. That triggers this process to block any other sperm from entering because all the receptors on this plasma membrane are then going to be kicked off. So once one sperm binds, the rest of the receptors on the plasma membrane disappear. So even if you have another sperm that shortly thereafter made its way through the zona pellucida, it now has nowhere to bind. That blocks polyspermy, which is multiple sperm fertilizing the same egg. So looking at this step by step, review this picture on your own. You have the acrosomal reaction of the sperm trying to burrow their way through the zona pellucida. That calcium influx is what triggered them to release those enzymes. Binding occurred when the sperm bound to the plasma membrane of the oocyte, so it had already dug through the zona pellucida. It fuses with the plasma membrane of the oocytes. Their plasma membranes join. And then to block polyspermy, you have this oocyte membrane block where those receptors disappear. And then finally, you have this cortical reaction where calcium is going to be released out of granules, these cortical granules in the oocyte, and that'll secrete calcium into the zona pellucida, and those receptors in the zona pellucida will also be destroyed. So a kind of three-step process: take the receptors out of the plasma membrane, calcify the zona pellucida, and destroy the receptors in the zona pellucida. So that's how we block monospermy. So polyspermy. We want in humans just monospermy to occur. So you block the membrane receptors in the membrane of the oocyte, and then the cortical reaction is when you are going to calcify and get rid of the receptors in the zona pellucida to prevent any other sperm from getting through. So what happens when one sperm does make it through? It then has to go through the actual process of fertilization. So its plasma membrane has joined with the eggs. Its nucleus is now just kind of like floating around inside of the oocyte. That triggers the oocyte to finally finish meiosis. At this point, it was still arrested in meiosis 2, so now it finally completes the last stages of meiosis 2. Its final polar body is just kind of like kicked off to the side. It'll degenerate later on, but then these pronuclei, the pronucleus of the male and the pronucleus of the female, start to swell, and they are going to replicate their DNA. They're still haploid organisms, but they need to make those sister chromatids so they can create an entire diploid organism that can then grow and divide and go through its own mitotic divisions. So in this process, they're meeting basically in the middle of this cell at these mitotic spindles, which kind of create a space for them to line up. And then their nuclear membranes are going to fuse. This is where you actually have fertilization occurring. This is the creation of a new individual, essentially. So you have a zygote, finally, is what we can call this structure. Before, it was just a sperm and an oocyte. Finally, you have what we can call a zygote. And that is when the DNA is basically being combined into one new individual. After this, you are going to quickly have mitotic divisions that we call cleavage. And I actually like your book's description of it. Your book says that you can have this giant granite block, like a picture of a huge boulder, but you can't build a building out of one huge boulder. You have to break that boulder into smaller and smaller and smaller bricks. With those individual teeny tiny bricks, you can create a building. So this process of cleavage is these really quick mitotic divisions, but the cells are just getting smaller and smaller and smaller because you don't have any like growth of the cytoplasm or anything. So it's just still contained in this same circle surrounded by the zona pellucida. But those cells are just going to divide and divide and divide and get smaller and smaller and smaller. So after about 36 hours, that first division has happened after fertilization occurs. And then about after that, 72 hours after fertilization, you have 16 cells or more. That is called a morula. So the zygote here, surrounded by the zona pellucida still, that fertilized egg, 72 hours later, you have this morula. And then later on, it is going to become a blastocyst. And at this point, it's still just traveling through the uterine tube. It finally makes its way into the uterus at about 4 or 5 days. We call it a blastocyst when you have a fluid-filled cavity in the middle here. And that blastocyst is where we have the first just basic stages of cell differentiation. On the outside, you have cells that are called trophoblast cells. And on the inside, you have the embryoblast cells. The embryoblast cells, as their name implies, become the embryo. The trophoblast cells do all the dirty work of implanting in the uterus and creating the placenta. So they have their own roles. They're actually going to secrete hormones and do all the other jobs. The embryoblast cells become the embryo on the inside there. The next thing after fertilization, we've made our way through the uterine tube. We now have to implant ourselves into the endometrium of the uterus. So mom has to supply all the nutrients, all the blood, get rid of all the waste as this embryo develops. So to get to that point where you can start having mom passing all the nutrients and stuff to and from, you have to implant in the uterus. So implantation occurs 6 to 7 days after ovulation. Remember, mom's endometrial lining, that functional layer, has been building up this whole time so that she can support this incoming blastocyst. And so by the 12th day, this implantation process will be complete. The trophoblast is going to secrete those trophoblast cells will secrete HCG, or human chorionic gonadotropin. That is what rescues the corpus luteum. So maintains the corpus luteum, or CL, because the corpus luteum is producing progesterone and estrogens, which prevent mom from spontaneously aborting or shedding that endometrial lining through menstruation. So you have to have HCG coming from the blastocyst itself, from those trophoblast cells, and you have to have this process of implanting into the uterus before you can start to grow and develop. So we've made our way. The egg has been ovulated, fertilized somewhere in the beginning stages of the uterine tube. All these cell division processes happened, getting to a morula and then a blastocyst with this fluid-filled cavity. Finally, we have our first beginning stages of cell differentiation to create our trophoblast cells and our embryoblast cells, and we're ready now to implant into the lining of the endometrium, which is the inner lining of the uterus. So, what does this process of implantation look like? Well, the zona pellucida is going to degrade. It's been still surrounding the blastocyst, supporting it, making sure it is not degraded in the uterine tube as it travels through and then floats around in the uterus. Once that degrades, these trophoblast cells can actually bind to the endometrial lining. So they can bind to the uterus and allow growth to start to happen. Once they bind, they're going to start to secrete these chemicals that start, it's essentially a parasite now, because it's going to start digging, eating its way into the endometrium of the uterus. We have these syncytiotrophoblast cells that are special trophoblast cells, especially for that process. And so you still just have the embryoblast hanging out in here, not really doing anything yet, waiting for implantation to occur. So this blastocyst invades the uterus, and we want the uterus to completely envelop this blastocyst so it can completely support, surround, and provide all the nutrients that this embryo is going to need. Those syncytiotrophoblast cells dig into the uterus and allow that to happen. And at this point, we have, and we'll come back to this bilayered embryonic disc. This disc here is what actually develops into a human. Everything else is just kind of structural and supportive and will become the placenta and the amnion, which is just the sac surrounding the developing embryo. So this embryonic disc here will become the little person. Everything else is developing from those trophoblast cells. So the trophoblast cells differentiate into this chorion structure. So all of these cells around here make up the chorion. The chorion is going to start to join up with mom's tissue, and the chorion and the endometrium together, part of the baby and part of the mom together, join to form this structure called the placenta. But at this point, we just have a few structures. So we have the amniotic sac beginning to form, which will surround the embryo as it develops, provide some support. We have the yolk sac, which in humans is not that important because we're in mom the whole time. So in creatures that lay eggs like chickens, the yolk sac provides all the nutrients as that baby develops. For us, it has a couple of roles, but it's not going to last in a major role for very long. The syncytiotrophoblast cells out here that are doing the digging into the embryo. And from here, we can start to develop the placenta. So implantation, this whole process, 2/3 of zygotes will never get there. So 2/3 of zygotes are going to spontaneously abort by the end of the first week because they fail to implant. After that happens, another 30% that implanted are going to spontaneously abort typically due to some genetic defect, but it can also be because we are not producing enough hormones of one type or the other, or the uterine lining was not fully developed to support that incoming zygote blastocyst. The corpus luteum, if it degenerates, again, probably spontaneous abortion. You can sometimes supplement if it's just a hormone issue and give a woman progesterone and estrogens. But if there's a huge, malformation of the uterine lining or of the blastocyst itself, spontaneous abortion will occur. How do you get twins? We kind of talked about this in the last chapter, but I'm just going to briefly cover it again. Identical twins are when you still had just a single egg and a single sperm that are going to, at some point in that process of becoming a blastocyst and becoming an embryo, are going to split in two. Fraternal twins are when you have two separate eggs fertilized by two separate sperm. Still very rare. But the only ones that are going to share all of their genetic information are identical twins because they developed from the same egg and sperm. You can have twins that share a placenta. It's not very common. And typically they also have their own separate amniotic sacs. So this is when two separate blastocysts implanted usually in two separate parts of the uterus. And so they're all going to develop their own placenta and their own chorion and those trophoblast cells, which will become all of those structures. So how does human chorionic gonadotropin come into play? You may know that this is the hormone that is used to detect pregnancy, but what is its role and why is it secreted so soon? It's basically the first hormone secreted to tell mom that a pregnancy has occurred. So it's going to be secreted by those trophoblast cells as they start to implant in the uterus to make sure that mom knows that a pregnancy has occurred and her body does not try to kick that blastocyst out, recognizing it as like a parasitic invader or something like that. So rescues the corpus luteum so that corpus luteum can continue to produce progesterone and estrogen. That makes sure that menstruation does not occur. It is going to also help development of the placenta. Those estrogens and progesterone are important for placental development. At this point, this crucial point is when the placenta starts to take over production of estrogens and progesterone. So this is the point where human chorionic gonadotropin has started to decline. And so along this same track, the corpus luteum is also going to start to degrade because HCG is necessary for the corpus luteum to exist. So once the placenta is ready to take over, human chorionic gonadotropin declines, stays at really low levels at the rest of pregnancy, but the placenta is going to take over producing estrogen and progesterone. So placentation is when you have implantation has occurred, those trophoblast cells develop into that chorion structure surrounding those embryonic cells, and that chorion is going to become the embryo part of the placenta. And then some of mom's endometrium is going to kind of fill in and surround that chorion structure and become the mom side of the placenta. We have to have mom's blood supply passing all of the nutrients. The embryo side of the placenta is going to pick up all of those nutrients without allowing a lot of intermingling of blood. We don't want that to happen because mom can start to recognize the baby as an invader. But sometimes there's a little bit of blood mixing, and that's usually just if part of this like chorion structure has maybe torn in some places. But for the most part, no blood actually mixes. The placenta is fully formed by the end of the third month. So that's when this occurs, this point where the corpus luteum can start to degrade as the placenta takes over that hormone production. The placenta is also the structure that provides all of the nutrients for the growing baby for the entirety of the pregnancy from the third month on. So producing progesterone and estrogens are really just to maintain the pregnancy, and then the placenta also does all of the hard work of passing nutrients and wastes to and from the developing embryo and then fetus. Those hormones that come from the placenta will also help prepare the mammary glands to start developing those lobes, which are going to be milk producing when you are late in pregnancy. If you don't have enough of any one of these hormones, again, you can have spontaneous abortion occur. So all of these hormones are necessary to maintain, neural communication with mom, to make sure that she is aware that her body needs to start to prepare and grow. The uterus has to grow a huge amount. So you have to have this communication with this hormone, all these hormones between mom and the developing embryo for a pregnancy to be successful. Placentation, or the development of the placenta, occurs as a combination of the chorion, which is going to develop these villi, those finger-like projections, into the endometrium, or the uterus of the mother. And then mom, her endometrium is going to start to fill in those lacuna, those spaces between the villi, with really a rich blood supply. So that is what really gets the rich blood supply going to prepare for this major, major growth that is going to quickly occur in the embryo. And in the middle, you can see we used to just have these 2 cell layers of that bilayer disc. We now have 3. And so we have these 3 germ layers that will produce all of the cells of the baby. So the ectoderm, mesoderm, and the endoderm. Before, we just had the endoderm and the ectoderm. This mesoderm was the last one to show up, but the mesoderm is actually what creates most of your body, like all of your skeletal muscle, all of your tissue, almost all of your organs are going to be formed from that mesoderm. Remaining, you still have the yolk sac, the amnion. The amniotic sac is not yet surrounding the embryo. We have to have placentation first. The placenta has to develop before we can start to really balloon out into the uterus and develop this growing baby. The yolk sac is still there, kind of left behind. This structure, called the allantoy, is going to form the umbilical cord. So this is just kind of like a pinch off of the yolk sac that will be left behind to communicate with the placenta, between the placenta and the embryo. So this is more developed. This slide was at 16 days. So really, just like a few cell layers are all that make up your embryo, just like a few sheets of paper stacked on top of each other, with just some cells. Finally, at weeks 4.5, so only a couple weeks later, you can actually start to see this structure that looks like some sort of animal. Every animal with a vertebrae looks essentially the same at this point. So you could look at a dog, a cat, chicken, a whale, they'd all look pretty similar at this point. But you can also see the placenta has really started to develop. So the chorion, which developed from those trophoblast cells, has started to intermingle with the endometrium, forming this rich blood supply between the two of them. Again, the blood does not mix. So these villi of the chorion will always just kind of like stay floating out there, and then mom's blood supply is just going to pass, all the nutrients will be passed between cell layers of that placenta, which becomes the chorion. Another thing that you can see at this point is that the amnion has formed a complete circle around the developing embryo. So we have the amniotic cavity finally inside here, and the yolk sac is just kind of a residual piece left on the outside. It has a couple of roles, but will never really develop any more than it has at this point. And that allantoy, which was a pocket coming off the yolk sac, is going to start the formation of the umbilical cord right here. So some of the blood vessels of the chorion dip into the amnion to kind of connect right at that umbilical cord. And the umbilical cord is what actually passes everything to and from the placenta. The placenta has, a mom side of it that communicates with the rest of mom's body and provides everything that the baby needs while it develops. We can then, after the placenta has developed, we had implantation, the placenta developed. Now this amniotic cavity can start to balloon and, grow into the rest of the uterus. We have these layers called the decidua capsularis on the outside, and extra embryonic coelom, which is just a space in between there and the amniotic cavity contained within the amnion. The yolk sac, still left over, but very, very small now compared to everything else. And the placenta, that chorion and endometrium of the mom, have developed and are going to stay in this same spot for the rest of the pregnancy. All right, so you can see closer up what this blood supply looks like. The chorion formed these villi, which are going to communicate through some thin cell walls with mom's blood supply. So blood never mixes to and from the baby. It's just these little finger-like villi. They're going to be projecting into spaces that are surrounded by mom's blood. All of the nutrients and gas exchange and waste from the embryo are going to be passed, across those thin cell walls, but they're never going to be mixing together. And then this is the start of the umbilical cord that travels from the placenta to the growing embryo. Okay, so now we can finally start to go through all of this differentiation and growth. A lot of this is happening at the same time as that placental development. So you have the 3 germ layers, the gastrula. The 3 germ layers are just called the gastrula. Those will start growing as the placenta starts to develop. So this is not really after all of that stuff. It's happening at the same time as the placental development. The amnion, which is that sac, which will form the amniotic fluid, that's what is going to rupture when a woman says that her water breaks at the beginning of giving birth. The yolk sac produces red blood cells, and that's one of its major roles right at the beginning of pregnancy, because the fetus does not yet have red bone marrow, right, which forms red blood cells in an adult living human. So the yolk sac is the first thing that is going to start to form red blood cells and allow the cardiovascular system to maybe start to develop from there. The allantois is just that pinch off of the yolk sac that will form the umbilical cord. And the chorion was developed from those trophoblast cells, which will become the embryo side of the placenta. So we just had that 2 bilayered disc at the very beginning in that blastocyst that had developed. Now we have a mesoderm. So this little hole right here is that first little bit of the mesoderm that was formed, which is the notochord, which told the baby, like which side is up and which side is down, essentially. The mesoderm will then hugely grow and produce everything in our body other than our skin and a very inside layer of our gut and some of our digestive and hormone glands. So looking at this a little bit closer, that notochord formed, and then the mesoderm continued to develop on the outside of that. At this point, the placenta has still not fully developed, so we just have that amnion, which is, this pocket here, and then the yolk sac, this pocket on the other side. The yolk sac will then kind of be moved aside. The amnion will surround everything, all of these 3 germ layers. But gastrulation is just the formation of these 3 individual germ layers: the ectoderm, the mesoderm, and the endoderm. The notochord is that first little bit of the mesoderm that is going to form to just tell us which way is up, which way is the brain, and which way is the tail, essentially. And then the endoderm is going to become the epithelial lining, so the inside of your gut. The ectoderm will become your nervous system and your epidermis, so the outside, your skin, and then the nervous system as well, while the mesoderm produces everything in between. And you can see how the mesoderm has to differentiate into all of these different tissue types that are going to differentiate into everything inside of our body, while the ectoderm and the endoderm really don't have much other further differentiation to do. You can spend some time studying this. I don't need you to remember all of the individual tissue germ layers that form off of the mesoderm. Just be aware of what their final products are, what these 3 layers provide. And these 3 germ layers are not, this is not just human development. Basically every other species of living animal on the planet looks like this at this point. So this is not special to us. This is just, we have evolved from species that came before us. So this is not something that is, just part of the human development, but we of course have a lot more DNA than, simpler creatures do, that allow us to have this further development of things like kidneys and gonads and a brain that is extremely developed with the spinal cord. So we will start to differentiate very, very quickly as this embryo starts to grow. But we have this circulation that allows all of this to happen, allows all of the nutrients, the amino acids, glucose, carbohydrates, oxygen, all of the nutrients that the baby needs, those building blocks of life, and then allows CO2, because that's still a waste product, even for a growing embryo. CO2 and all the metabolic wastes that are produced by the growing embryo pass the other direction out of the umbilical cord. So when we're talking about umbilical circulation, we usually say arteries go away from the heart. In this case, it's the same thing. You're just talking about going away from the embryo. So the umbilical veins go towards the embryo and carry oxygenated blood from the placenta. They will pass through the liver a little bit, because the liver is what does the initial processing of all of our nutrients and things. The umbilical arteries will carry deoxygenated blood, because the embryo is in a sac of fluid, right? It is not breathing air into its lungs. So it needs to have oxygen and all the nutrients coming from the umbilical cord, coming from the placenta. So umbilical arteries, deoxygenated blood; umbilical vein, oxygenated blood to the embryo. We have these vascular shunts. The ductus venosus is going to, for the most part, bypass the liver. We say bypass, but of course there is still some blood going to these structures, because you have to have enough blood going to them to help them develop. But for the most part, it just dumps into the inferior vena cava, bypassing the liver. So this will occur down here. And then we have structures in the heart as well, the foramen ovale, which is between the two atria, because again, you don't have developed lungs. And so all of these other structures with the ductus arteriosus, these are going to help to bypass the lungs, because the fetus is not yet breathing oxygen. So the ductus arteriosus, let me see if I can draw this for you a little bit better. The ductus arteriosus is going to dump blood from the pulmonary artery coming off of the right ventricle that would normally go to the lungs, and it is going to dump most of that blood into the aorta. So instead of the pulmonary artery taking blood to the lungs, it's primarily going to just pass it out into the rest of the body via the aorta, and then it can go into the umbilical circulation in the umbilical arteries and be, pick up our oxygen and everything there, because the lungs don't have oxygen. So you can take a look at this. Your book has this really great picture just to orient you. This is the placenta down here with the umbilical cord. You cannot see the baby in this picture. So the baby is just like outside of all of these structures here. This is the baby. It's kind of confusing at first when you look at it. So this is the developing fetus, and these are what these structures look like in a newborn baby. So the placenta is still, attached to the endometrium of the uterus, but you have all of these special umbilical veins and arteries that are going to bypass the baby's lungs and allow oxygen and nutrients to come from the placenta. So pay attention to these, because all of these are going to be important for those processes, and they're all going to essentially just close off after birth. Once the baby takes its first breath, it can then, pass air in and out of its lungs. And so the ductus arteriosus and the foramen ovale in the heart are going to close, basically with the pressure of just like blood pushing back against them, and then they will close completely like a few weeks after birth. The umbilical veins, you're going to pinch off and cut the umbilical arteries and veins, and so no blood will even be flowing through those. So those will just kind of like naturally come shut and close on their own. So this is where this red one right here is that umbilical vein, and these purple ones coming off of the superior, off of the abdominal aorta, are all going to just naturally close on their own, all of those umbilical arteries and veins. So this is what a developing fetus looks like at about 7 weeks. We have the yolk sac kind of off on the side here. The amniotic sac is now surrounding, and the chorionic villi are communicating with the endometrium of the mother. Your book has a really good table that includes descriptions of what is happening. There's obviously a lot happening at all of these stages, so I'm not going to go through it all. At about 12 weeks or month 3 or so is when you have the fetal heartbeat begin. So a lot of the organs at this point are already discernible and have started to develop and become functional. Obviously, you have an extensive amount of brain development that has started to occur here. And you can see the hands and feet, eyes starting to form. So every structure has started to develop at about this point. And at about 5 months, you can see most of the other structures have developed. At 5 months, your kidneys have started to develop, bones are discernible, the eyes are starting to become functional, like can blink, and so you have some of your sensory organs developing. You can see some of those other stages in the fetal development process if you look in your book, but obviously you can see this drastic growth that happens during just, a few short weeks. Really, 9 months is not that long. And so you can start to get these structures appearing very early on. Between 6 and 12 weeks, a lot of the organs are starting to develop, and then they just differentiate and become functional from there. Now we're going to look at the mom side of things. So obviously, as this fetus is developing, become from an embryo to a fetus, starts to grow really drastically, a lot of changes are going to be occurring in the mother's body. Not all of them are going to be good. Some women say that they have, like a great pregnancy, they love being pregnant. It's not that way for everybody. There can be a lot of complications and a lot of just, general wear and tear on mom's body because she has this embryo and then a fetus like ballooning inside of her abdomen that is pushing on all of the other organs, pushing her diaphragm up, pushing her stomach up into her esophagus. So you have gastric reflux and a lot of other things that can be occurring as the baby develops. So Chadwick's sign, this is when you get purplish hue in the vagina, and that's pretty normal. You just have this deep vascularization occurring. Breast changes, of course, as those lobes start to develop and be able to produce milk. Chloasma is when you have this pigmentation in the skin. That's pretty typical. Uterine growth, of course, has to occur probably more than anything else, as that endometrium develops into the placenta and then grows to expand to contain the fetus as it grows. But then that myometrium, those muscle layers outside of the endometrium, have to grow and develop as well, because those are what are going to force the fetus from the uterus and expel it out of the body during birth. Relaxin is this hormone that is produced by the placenta. So this is just another one of the hormones that the placenta produces. This is going to soften the pelvic ligaments and widen the pelvis to allow that birth canal to develop and to, allow a woman to give birth naturally. If you don't have enough relaxin, or you have some other anatomical abnormality that doesn't allow this process to occur normally, you probably will have to have a cesarean section or a C-section, because if those bones are not widening and the ligaments are not loosening enough, that can be extremely, extremely dangerous for the mother and for the baby. Weight gain is very normal. Of course, you are holding an infant. The infant only weighs a few pounds, but you're also adding a lot of water into mom's body, because she has to have, increased blood flow to accommodate for getting all the oxygen and nutrients and waste to and from the growing fetus. So weight gain is pretty typical. A lot of that will be lost after the pregnancy. This is what those face changes, that pigmentation looks like in a pregnant woman, fairly common. So what does this look like in the breasts and in the uterus as this occurs? So, 4 and 7 months in, that uterus is the most obvious, not from the external portion, obviously, but that's when that embryo is starting to develop extremely quickly. And then the breasts around 7 months are going to start to develop as well, as that glandular tissue grows and accommodates for, milk production to happen. A mom is not able to produce milk until about 9 months of pregnancy, however. So that's not going to happen too early on. You don't need it until about 9 months of pregnancy, unless you have a premature birth. The GI tract, heartburn and constipation, all of these things are really just because the uterus is expanding so much to accommodate for that growing fetus. And so this is pushing on all of the other organs. Morning sickness is typically due to elevated progesterone levels. You urinate more because the baby is pressing on your bladder, but there are physiological effects that cause that as well. Respiratory changes, so your diaphragm is being pushed up, your lungs are being kind of compressed, that whole thoracic cavity is going to become smaller, but you also have an increased tidal volume, which is going to occur because you need to increase your cardiac output. You have more blood volume in a pregnant woman because there's more water in the blood. And so you need to have a higher tidal volume to make sure that you are pumping that higher volume of blood around the body. This is more so, however, due to the fact that you have an increased production of CO2, especially because you also have a fetus producing CO2 at this time. Dyspnea can occur, which is just trouble breathing, and that's because you have an increased tidal volume, but also because the baby is pushing up on the diaphragm. The cardiovascular effects are the most pronounced of any of these effects. So you have a drastic increase in blood volume, 25 to 40% higher blood volume by 32 weeks. So that's pretty insane. So that is what's going to cause your increased cardiac output. To make up for that, make sure you're pumping that blood out at a pace that you need to. If you have impaired venous return because your femoral vessels are being pressed on, all of those vessels going into your legs are going to be kind of squeezed as the baby grows. And so that could impede your venous return, and that can cause varicose veins, which are very unpleasant. Something to maybe take into account is if you are active, your baby will likely weigh more. It's probably due somewhat to an increased metabolism because your baby is going to recognize whatever the environment in the womb is and kind of like match that with growth a little to a small extent. Okay. Preeclampsia. This is an extremely dangerous condition. It is diagnosed when you have high blood pressure, consistently high blood pressure, and if you have protein in your urine. Protein in your urine can lead to edema because you have more solutes staying in. When more solutes stay in your urine, that is going to make fewer solutes remain in the blood, and proteins in the blood are what help us maintain blood volume. But hypertension can lead to all sorts of issues. Full-on eclampsia is when a woman starts to have seizures that can be fatal to her and or the baby. The only way to cure preeclampsia or eclampsia is to have the baby. That can be, of course, dangerous if preeclampsia occurs too early on in pregnancy, because giving birth to a premature baby has all sorts of issues on its own. So that's why doctors are always, regulating blood pressure and making sure that we're monitoring blood pressure regularly. The initiation of labor. So this is going to be brought about by triggers from the fetus. So the fetus is going to control most of this because the fetus essentially knows when it's ready to be born. This is going to be caused primarily by an increase in corticotropin releasing hormone from the fetus, which will then send signals to the mom for all of these other things to happen. So fetal cortisol is going to rise, and this is a positive feedback loop. So this is another one of those rare cases where positive feedback mechanisms are occurring. So you have this continual circle of the fetus releasing corticotropin releasing hormone, which causes an increase in cortisol. Again, increased CRH to feedback and produce more cortisol. This happens just near the end of pregnancy, getting close to birth. Increased cortisol is going to cause higher levels of estrogens and lower levels of progesterone. Progesterone is what maintains the hardened cervix, so preventing the cervix from becoming too loose and allowing a premature birth or, a spontaneous abortion, that's why you have high levels of progesterone, or one of the main reasons. So when progesterone declines and estrogen gets higher, you start to secrete these things called prostaglandins, which will help to loosen the cervix and stimulate the uterus to contract. That leads to stretching of the cervix. When the cervix itself stretches, that causes a release of oxytocin from mom's posterior pituitary, and that is going to lead to another positive feedback loop. Again, you have 3 places of positive feedback loops here. Stimulation of contractions at the uterus will cause more prostaglandins, which will cause greater amounts of stretching, which will then lead to oxytocin to be released. And oxytocin is what actually causes the myometrium to contract. So this all stems from fecal corticotropin releasing hormone, which is fetal corticotropin releasing hormone, which is kind of an odd way to start this whole process, but it's because it leads to this chain reaction. When labor starts to occur, the first thing that happens is this dilation stage. So this is when the cervix starts to dilate. Remember that the cervix is not the vagina, right? So the doctor has to, you always see in like medical shows, feeling like how many finger widths apart. When you get to 10 centimeters dilated, you can say that you are like fully dilated, and it takes about 6 to 12 hours to get to that point, which is why Pam in the office waits so long in her second pregnancy to go to the hospital. It's not like it's urgent right when you start to have contractions. It's going to take you a long time to get to this point. Engagement is when the baby is actually moving into the birth canal, so actually has contact against the pelvis, which can, cause these greater contractions to start to occur. But the reason that we have this dilation occurring is because the head of the baby is actually pressing on the cervix. So that's what's actually causing the cervix to expand as the bones of the pelvis start to expand as the baby presses up against them. So dilation is when the baby's head is engaged with these pelvic bones. That starts that whole process. These contractions start. They are, when they start, they are not as strong, and they're going to be further apart. Late dilation is when the baby's head is in the widest part of this cavity, and then you can start to move into the next phase, which is expulsion. So that is when the baby's head is going to be delivered. This process is going to be much, much shorter than the dilation period. So in less than an hour, almost always, from fully dilated, you will actually have the baby. So expulsion, typically about 50 minutes if this is your first time birth, can be as 20 minutes or shorter in any subsequent birth. So if you have had a child before, this process is usually a lot faster. But this is what causes the urge to push. Crowning is when the head is actually the widest part of the head is visible in the vulva. Vertex or breech positions are just these extremely dangerous birth positions that the baby is in that can cause a lot of issues to go wrong. But then the placental stage is when the placenta, about a half hour or so later, is going to be delivered. Uterine contractions are going to continue, which are just going to sever the placenta from the wall of the uterus. And then the doctors can actually count the number of umbilical veins and arteries in the umbilical cord to make sure that the fetal circulation has developed properly, to make sure that the baby has all of the vessels that it needs. They can actually count some of these vessels in the umbilical cord. So placental stage happens about 30 minutes later. This is what you call the afterbirth. And so all of the placenta with all of the fetal membranes are going to be expelled at this point. Contractions continue to help that happen. If you have a baby that is, pressing up against its placenta, up against the cervix, that is called placenta previa. Obviously, this is very dangerous because you can have a lot of bleeding occur. And if, if the birth process does not happen fast enough, the baby can go too long without oxygen. And so this is extremely dangerous. The placenta should be somewhere up higher on the uterine wall. Preeclampsia is that condition in the mother, which causes, high blood pressure, proteins in the urine, can result in seizures. A breech birth is when the baby is not coming out head first. So complete breech is when it's folded in kind of this pretzel position. You have all these other breech positions. These are all very dangerous and can all be very painful for the mother to give birth naturally, because this is going to make it much harder for the baby to exit the birth canal. The placenta can also be, the umbilical cord can be potentially wrapped around a baby's neck, which can suffocate the baby as it's being born, as that placenta, as the umbilical cord is pulled. If there is a knot in the umbilical cord, obviously all of these things can cause major issues. And all of those can be a reason for a cesarean section or a C-section. After the baby's born, assuming all went well, we have this neonatal period of 4 weeks after the birth. And we, right away after the baby is born, within the first 10 minutes or so, we test these 5 signs: heart rate, respiration, color, the baby's actual color, muscle tone, and reflexes. Each of those are given a score with a maximum of 2 for a maximum of 10 points. If you're around an 8 to 10, that means the baby is healthy and good to go. Anything less than that could cause, doctors to take action for, potentially one of these things not being developed properly. Maybe an organ is not developed properly. So that's called the APGAR test, named for Dr. Virginia Apgar. It's kind of a shock to the body for the baby, because it has been in the womb for 9 months, developing, never breathing air, getting everything it needs from the placenta, from the mother. So this first breath can be extremely painful because you have to expand the lungs. So I always think of somebody that has had a pneumothorax, had a punctured lung, and how painful that is for them. Definitely not a treat for a baby either. But CO2 starts to accumulate because it cannot get rid of it through the umbilical cord anymore. So that acidosis triggers respiratory centers in the brain to trigger that first breath to be taken. The airways have to fill with air, and then the lungs have to expand. A lot of times this collapsing and expanding can happen kind of over and over as these try to start to develop on their own. Surfactant, which is that fluid that allows the lungs to stay inflated, has to be produced. A premature baby cannot produce its own surfactant, because this is, the development of the ability to produce surfactant happens in about the 9th month of development. So if a baby cannot produce its own surfactant, it probably has to be on a respirator for, days or weeks until it can produce its own. There's this thing called the transitional period, where the baby is going to start to regurgitate, and as its airways open, as all of these pathways through its body that have never been exposed to the outside world work for the first time, this period of instability happens for the first 6 to 8 hours, and the vital signs can kind of go back and forth a little bit until it becomes more stable. Lactation, of course, is a very important thing to occur, because mom is going to pass a lot of important nutrients, especially in that first few weeks, to the baby, pass some of her antibodies and other immune cells to provide the first amount of immune system health to the baby, and also all of the nutrients that the baby needs. So prolactin is this hormone that is going to be rising throughout pregnancy, and it is what will stimulate the development of the ability to produce milk. It is going to fall after pregnancy has ended, after birth has occurred, if the mom does not breastfeed. So suckling behaviors are what cause this to continue to remain high as long as the mother is breastfeeding. Colostrum is what is produced in the first week following the birth, and it's this yellowish fluid that doesn't actually contain a lot of lactase or lactose, but it has a lot of proteins and a lot of minerals, and it still has some antibodies and other important immune response molecules. So the letdown reflex, this is a positive reaction, feedback reaction, where oxytocin is going to expel milk, and that suckling behavior is going to cause a further increase in oxytocin. So that positive feedback mechanism that allows this continual reaction to continue to eject milk out of the nipple as the baby is suckling. And that suckling behavior will also lead to continued elevated prolactin. So these mechanoreceptors in the nipples are going to stimulate prolactin to be released from the hypothalamus. So anterior pituitary secretes prolactin into the blood. That is what results in milk production. Oxytocin is important, also going to come about from the suckling effects, just that mechanoreceptor in the nipples, which is going to tell the posterior pituitary to release oxytocin. And that is going to be important for milk letdown or actual milk ejection from the nipples. So you have to have both of these hormones present. Positive feedback is going to happen just due to oxytocin, but you have both of these increasing as mom is breastfeeding throughout the first however long she wants to. Once you stop breastfeeding, these things will naturally start to decline on their own because you no longer have these mechanoreceptors being stimulated. So breast milk is not absolutely required. We have a lot of good formulas out there that you can, if you need to, use. There's just a lot of stuff in breast milk that is good and obviously extremely natural. So there's a lot of fats and irons, amino acids, all of the building blocks for life, and then all of these other chemicals as well, like the immune chemicals. So you probably recognize a lot of these. IgA, which is one of those antibodies, complement, lysozyme, interferon, all of those that help in the baby's immune response, because whatever mom has been exposed to, she's already producing antibodies for whatever is in her environment. So this is a really good way to provide some early stages of immune functionality, which the baby is probably not prepared yet to produce on its own. Prostaglandins prevent an immune response, an inflammatory response, along with interleukins. Glycoprotein, which prevents this ulcer-causing bacterium, that Heliobacter pylori. So that's that bacteria that if it gets into the gut, it will burrow into the stomach and cause all those gastric ulcers. So mom helps prevent the baby from that issue, providing good bacterial colonization by providing the sugars that bacteria in the gut will like to feed on. So you need that good flora in your body, and then natural laxatives, which help the baby to, expel all the waste from the body until it can actually have proper food and nutrients, which are producing its own fecal material. We have ways to assist in reproduction if a mom and a dad cannot produce a baby on their own. And this is known as, in general, reproductive technology. You've probably heard of artificial insemination and sperm donors and oocyte donation. A lot of times parents want to use their own egg and sperm. And so we can do that. And artificial insemination is simply when you are going to probably take a sperm from dad and some eggs from mom and fertilize them in like a petri dish type of thing, and then replace them into mom, either in her uterine tubes or somewhere in her uterus after that fertilization has occurred, and hope that it correctly implants in the mother's uterus. These techniques have gotten a lot better over time, but unfortunately, they're definitely not 100% successful. And a lot of times couples end up spending a lot of money and maybe don't ever have success. So there are other ways, like a surrogate mother who can carry, who can provide an egg, and you can still use the dad's sperm. There are a lot of ways that you can do this. Embryo adoption, if the sperm are used to artificially inseminate another woman, if the egg is the problem. So it kind of depends on who is the issue. Is it dad's sperm? A lot of times dad's sperm are not just motile enough. And so you can actually fertilize the egg outside of the body because the sperm can't get up there on their own, and then put that fertilized egg back into the body and hope that it takes. So hopefully this all kind of made sense to you. Review those steps of implantation and placentation. That's kind of the trickier things. And then once you get to the end stages of actually going through the birth process, all of that, the reproductive stuff that happens in the mom, all of that will become a little bit easier over time. And after this, you only have one chapter left, and that last chapter is really, really short. So hang in there. You're almost done. Let me know if you have any questions, and have a good rest of your day.